Hello everyone, my name is Anthony Bui and I am Group 6. For this oral presentation assignment, my group will assign with the RNA interference technique. By definitions, RNA interference is defined as the double-stranded RNA mediated interference, and it is the rapid method for silencing gene expression in organisms. As a result of this process, we gonna obtain the phenotype of organism is gonna be either identical genetic no mutant or just a series of mutants. So how does it work? Well, let's imagine that we have a short RNA molecules that inhibit gene expression, right? By triggering the degradation or translation. Or it can be inhibit by the transcription of the gene coding of a specific mRNA. So RNA interference really helps to mediate the resistance of both the endogenic parasitic and exogenic pathogenic nucleases and double-stranded RNA. It's also helped to regulate the expression of protein coding genes. That's the reason why RNA would been applied in many fields and used in many biological experiments, such as the therapeutic interventions, functional genomics, agriculture, and many other fields. So from there's two primary research articles that I found. The first one that, as you can see, I have figure one on the right hand side that I got from the article. The scientists were helping us to explain how the RNA interference pathway work for the gene silencing. Well, first of all, we have the for the endogenous microRNA pathway. They begin with the polymerase II transcribe primary the microRNA in the nucleus to become the pre-microRNA. The next step, the pre-microRNA gonna be moving to the cytoplasm and become a functional microRNA. The function of this particular microRNA is to inhibit the translation of the 3-gram untranslated region of the targeted mRNA according to the incomplete Watson-Crick base pairing principle. At the same time, we have the small interfering RNA enter exogenously to the cell and loaded to the middle region, as you can see right there. It's called the RNA induced silencing complex, or RISC. That's what I call. So for this RISC region, they help to guide degradation of the target transcript when a base pairing occurs. And at the same time, the small interference RNA is unwound during the RISC assembly. The single-stranded RNA hybridize with mRNA target, and the small interference RNA can also have to trigger transcriptional gene silencing by interact with the chromatin because they guide histone and DNA methylation to inactive chromatin. So all of these pathways in order to help regulate the gene expression. For the second article that I found, scientists would question about whether DNA-based drugs and RNA interference drugs are really stable drugs in gene therapy. Well, the answer is yes, because they allow a single treatment of viral vector delivered by short hairpin RNA genes. So for the figure 2 on the left-hand side, as you can see, the left side is the RNA-based drug, which they have the variety of targets delivered in vivo, and the right-hand side is the DNA-based drug, and the entire mechanism is regulated by DNA-based polymerase III or polymerase II enzyme, promoted the single hairpin RNA expressions. So these short hairpin RNAs can deliver viral or non-viral vectors to the target tissue as lentiviral. So the strength of this study is that they provided the strong target gene suppression without any toxic effect in gene therapy. But the drawback is something the scientists need to consider as well, because they're using a large number of animal model in this study, and they also showing the sign of off-target effects. Also, they also observed competition with cellular RNA interference component during the delivery methods in vivo. So the question I have for you guys, after you guys understand what the RNA interference technique is and how they can apply in many different ways, is that possible to determine a short hair RNA from single cells? Lastly is the reference of the two primary research articles that I found. Thank you. Have a good day.